In this lecture, we're going to talk about mostly Gregor Mendel and his discoveries as a geneticist working on pea plants. So he is from the Czech Republic, and he worked from around the mid-1850s. And he was mostly looking at one particular species of pea plants, and he wanted to create a mathematical principles which he could use to determine how inheritance worked. And he did all of this without ever knowing what genes were, or what a chromosome is, or what DNA was, or any of that stuff, which is, pretty fantastic. It's a really interesting and cool thing. His basic ideas were that there are these units of genetic material that come in pairs. And these elements don't change even over many generations, so those alleles, you could call them, or those genetic units are relatively stable and within a population. And also that pairs separate during the formation of gametes. And we're going to talk about all of those later. So he was looking at pea plants. This pea plant in particular had these properties such as that they could self-fertilize, that they can germinate, and that they develop relatively quickly, which made this a perfect plant to study because you can get in a whole bunch of generations in a short period of time. Like I just said, this life cycle allows for rapid generations, cross-pollination, and self-pollination. So you can take, where everything is. You can control this. You can take pollen from one plant and know which plant you're using to fertilize what. There's a wide range of characteristics that he was looking at, each of which had several varieties, which are called traits. And he got kind of lucky because genetics is actually a little bit more complicated most of the time than what he was looking at. Or not most of the time, but a lot of the time it's more complicated than what he was looking at. But he got lucky. He picked a model system, that pea plant, and he picked a bunch of characters that are easy to look at and very often follow Mendelian genetics. So he was looking at seed shape, seed color, pod shape, pod color, flower position, and stem length. And I haven't really read up on this, but I'm kind of realizing now he probably looked at more than these, but these are the ones that performed well, that made sense for him in his study. So before we dive in, I kind of wanted to be able to differentiate for you guys the difference between phenotype and genotype. Phenotype is the physical characteristic. So for instance, I have blonde hair, and so that's my phenotype. Phenotypes they can usually be readily observed. You can just look at someone and say, oh, that's your phenotype. Genotype, on the other hand, is the gene that determines the phenotype. So you can't just look at someone and just say, oh, well, that's your genotype. It's a more difficult situation, as we're going to find out later on. So he set up with true breeding crosses. They're called true breeding because they always give you the same kind of offspring. So if you have a plant that's true breeding yellow seeds, it's always going to give you yellow seeds. If you have a plant that's true breeding green seeds, it's always going to give you green seeds. And the starting generation is called P for parental. And he took pollen from one variety and placed it onto the stigma of another variety. So he took true breeding yellow and crossed it with true breeding green. The offspring are called F1 for first familial. You don't have to worry about why it's called F1, but you can just know P and then F1. And these kind of techniques are still used very regularly today, where you have a parental true breeding set, and then you're crossing them to create these F1s, or first filial generation. So to determine where green went, he did this F1 generation, and he found that all were yellow, even though he was crossing yellow and green. To figure out where that green went, he took those F1s and he crossed them back. He self-pollinated them. The next set of generation, or the next generation of pea plants, green came back, but in a proportion of about 3 to 1. And these are These are his actual numbers in his F2. 6,000, roughly 6,000 to roughly 2,000, which is really, really actually very good. So he was able to use these kind of crosses and determine what we now call dominant and recessive by looking at the F2 generation. Remember, P are true breeding things, so you would have ones that are always smooth or always wrinkled or whatever the trait is. And then F1 is the cross of those two true breeding things. F2 then is you take the result of those F or parental crosses, you take the result of those F1s, you cross those back to each other, and you get this 3-to-1 ratio of these things. And as you can see, it's always pretty darn close to 3-to-1. And later on, hopefully, this will make a little bit of sense to what's going on here. We're going to do some Punnett squares. We're going to figure these things out, why they are this way ourselves. So unlike his predecessors, Mendel was very, very careful, very, very systematic with what he was looking at. And he found that varieties did not blend. One is always dominant and the other one is always recessive. And those two traits he called alleles. So what we're talking about here, in this case, if you have a smooth seed and a wrinkled seed, each of these you would call an allele. So you would have a smooth seed allele, you would have a wrinkled seed allele. And in this case, he found that they didn't mix. So you couldn't have some combination of smooth and wrinkled, like halfway wrinkled or whatever. And that's one of the reasons why he was very lucky, because as we're going to see later, some genetic crosses do actually combine. So he called those different varieties alleles. In the F3 generation, 2/3 have mixtures of green or yellow, which were, excuse me, and 1/3 were all yellow. What's going on here is some things are being covered up by other things, and those are referred to as dominant individuals. And we're going to talk about this notion of homozygous versus heterozygous here in a second. So homozygous means you have 2 copies of the same allele. 2 copies of the same allele. So if we go back here, maybe you have 2 copies of the smooth seed allele, or maybe you have 2 copies of the wrinkled seed allele. Then you have heterozygous, which means you have 1 of each. So homo meaning same, meaning the same kind of alleles, hetero meaning different kind of alleles. And we're going to talk about how to use a Punnett square. So you take your true breeding yellows, and you take your true breeding greens, which it's safe to assume are homozygous. They've been crossed enough times that he felt fairly confident these were homozygous individuals. And he crossed them so you can figure out what the genetic results of your cross are going to be using a Punnett square. You take one set of alleles, which on the left here are big Y, and you put them on the left. You take another set of your alleles, which are little y, or homozygous recessive, and you put them on top. Then you mix those things together and you see all the possible combinations that you can get. And at the end, you wind up with, in this case, we cross 2 homozygotes and we round up with all heterozygotes. So you'll notice here that this is a yellow seeded pea plant, even though it has 2 different copies of our alleles. It has one big Y and one little y. And the reason for that is because the big Y is covering up any effect that the little y might have. A very common example of this is people's eye color. So if you have brown eyes, there's blue underneath there, but just imagine you're drawing eyes on a piece of paper with a crayon. And you draw in blue, but then you take a brown crayon and color over it. So you've colored over with your crayon, and now everything just looks brown, right? You can't see the blue anymore. That's what's going on here. This big Y is just overtaking this green color. You can't see any green left because the big Y is what's called dominant, or this big Y has complete dominance. So again, you can use these things called Punnett squares in order to determine what the various offspring are going to look like. So you take and put one set of gametes or one set of alleles on the left, You put one set of alleles on top, and then you can determine what all of your crosses are going to look like. So here we have a homozygous recessive. Here we have a heterozygous individual. Think about this. I'm going to let you fill this in on your own. What would you wind up with here? I'll just do it for you. What would you wind up with here in this square? Well, you would wind up with a big P and a big P. Okay, so that is homozygous dominant. What would you wind up with here? Well, you would wind up with the big P from here and the little b from here. So that would be a heterozygote. And then this one's kind of given away because you know what this one looks like. This is a heterozygous recessive. Right? You take all of your genotypes, and this allows you to figure out what the possibility of the offspring are going to look like. So on to Mendel's laws. We have this idea of segregation of alleles. So alleles you can kind of think of as trait. Pea plants contain 2 copies of each gene. Those individual copies are called alleles. Remember, you got one set of alleles from your mom, you got one set of alleles from your dad, and so you have these 2 different copies of these alleles. Alleles do not blend because in this case we're talking about complete dominance, and so one copy is going to mask the expression of the recessive copy. And alleles must separate during meiosis, and Mendel had some ideas about this, even though he had no knowledge of what chromosomes were or what meiosis even was. So the F1 to the F3 generation, here's our F1. We've taken and we have crossed them, and you can see you wind up with 3-to-1 yellow peas to green peas. You take these and you cross them again, and you get this F3 generation. And you can use your Punnett Square to figure out exactly what it is that these are going to look like, and you can hopefully kind of figure out why. So if you take this yellow seed and cross it to itself, you wind up with only yellow because those are homozygous dominant. If you take these heterozygous individuals, remember heterozygous means you have one big Y and one little y. They're mixed, hetero, different. And you cross them, you're going to wind up with all different kinds of individuals here. You're going to wind up with some heterozygotes like here and here. You're going to wind up with some homozygous recessive like here, and you're going to wind up with some homozygous dominant. If you take the green individuals, and make a cross out of them, you wind up with all homozygous recessive, you wind up with all green seeds at the end. So chromosomes, these are our chromosomes here, and they actually have a copy of each of these alleles on them. So you can imagine this one, you got this one from your mom, this is your maternal chromosome. You got this one from your dad, this is your paternal chromosome. This is a heterozygous dominant because you have 2 copies, both of which are the dominant version, and you're always going to wind up with yellow seeds from that. If you have another organism that has a heterozygous, this is a heterozygous individual, and they have one yellow copy and one green copy, you're also going to wind up with yellow seeds. Because remember, that yellow copy is the dominant one. It's going to overtake the green, and it's going to be yellow no matter what. No matter what that green one tries to do, it doesn't make a difference, yellow seed at the end. If you have homozygous recessive, then you're only going to have green seeds. And you can tell that it's homozygous recessive immediately because it is green. So if you just were to look at this yellow seed down here, would you be able to figure out its genotype? These are genotypes, by the way. These are the literal sequences that are making things up. So genotypes are these. Phenotypes are the color that the actual seeds are. So if you had a heterozygous genotype, you wouldn't be able to tell that just from looking at the seed. So again, you have, like I was just saying, 3 genotypes yielding only 2 phenotypes. Homozygous dominant and heterozygous are only leading to yellow. So even though these have different genotypes, they have the same phenotype. Whereas this one, you can tell immediately what the genotype is because it has this green phenotype. How do you know it's going to be green, or how do you know it's going to have two little Ys? Well, because if it had a big Y, it would be yellow. If it had two big Ys, it would be yellow. So you can tell immediately from this recessive individual. So we have this thing called a dihybrid cross, which I'm going to probably get into later. I might just leave it alone, we'll see. But we have this thing called independent assortment, which we learned about in the meiosis lecture. So gene pairs assort independently, and because of this independence and this randomness of the way they sort out, you can wind up with different kinds of these dihybrid crosses. And so, by the way, a dihybrid cross is when you take two traits and you try to look at two traits at the same time instead of just looking at one. And a dihybrid cross results in these big Punnett squares that look something like this. And you can wind up with all these different and kind of interesting combinations of things because, again, independent assortment makes them this way. I think we'll do an example in class this week just to make this thing crystal clear. But this is a dihybrid cross. So you also have things that are a bit different, like incomplete dominance. So we had complete dominance. Those were all the Mendelian traits. You have incomplete dominance. And with something like this, if you take a red flower and a white flower and you mix them together, you wind up getting pink flowers as well. Because genes make proteins, the red gene makes the red pigment. And because there's not enough red pigment, because you have that white allele, you wind up with only half. And so you wind up getting a phenotype that is a mixture of the two. So complete dominance, it's one thing or the other. Incomplete dominance, it is a mixture of the two things, a blending, I should say. So red and white, you wind up with pink. And this is just an image of that. So you have this dominant red and this recessive white. If you cross those together, you wind up getting some red, some pink, and some white. So you wind up with another phenotype at the end. We also have something called codominance, and this is going to be a question on the test that I'm going to ask you. And this is ABO is a good example of this. You have multiple alleles, multiple phenotypes, and genotypes of all possible combinations of A, B, and O. And I have a picture of this that I'm going to show you. So if you have an A genotype, you're going to wind up with an A phenotype. And you wind up with all of these surface proteins. These are actually called antigens. The word antigen is not important here. But you wind up with these antigens that allow your body to identify, hey, that's my blood cell. I know what that is. So you have this A blood cell here. It has all these little markers, these little antigens on its surface. You have a B blood cell here that has all these little B markers on the surface. But you can also wind up as an AB individual. So this is this idea of codominance. You're winding up with a mixture of both phenotypes. You have some A's, you have some B's. These didn't mix together and form like a C or something like that. That would be incomplete dominance. But this is codominance. You wind up with both. And you can also, interestingly, wind up with O, which is where you just don't have anything on the surface of your cell at all. The next topic is this idea of polygenic inheritance. You can pause it and go back if this is too much. But polygenic inheritance is one trait that is determined by many genes, and you wind up getting continuous variation this way. So Mendel's pea plants, they were either green or red pea pods, right? Green or red, there's no middle ground. For a polygenic trait, you're going to wind up with a whole wide variety of different traits, of different sizes. So here is a bunch of people. I think this is the girls, or the girls are in white and the boys are in blue. And you can see that there is a very wide range of variations. In fact, you could even go deeper into this, and you could go to the half inch, the quarter inch, the 1/10 of an inch, and get an even wider distribution of people. All right. So polygenic inheritance is a trait that is determined by many genes. In fact, most traits are influenced. Most traits are determined by many genes. You also have environmental determination. So hydrangea can change color depending on what soil they're grown in, what the pH of the soil they're grown in. And identical timberline plants will grow differently to different heights at different altitudes. So that's all I have to say about Mendel for now. Definitely ask me questions about this during lab. We're definitely going to be talking about all of this stuff in lab as well.